Hey everyone. If you're missing school for whatever reason, today we're going to be doing a 45-minute biology genetics makeup lesson.
This is Lasseter's Lab, your go-to resource for life science and life lessons, especially when you're reviewing for exams. In this video, we're going to review some important key content, do a little background mini lesson, and then have plenty opportunity for practice questions and a little bit of classwork from the comfort of your own home.
Give this video a like if it's been helpful for you, and be sure to share it with a friend if you think it could help others. Let's get started.
So this is Gregor Mendel. He's the father of genetics. So he determined that a single gene could have two different alleles or versions, such as a dominant and recessive version, and those different combinations could lead to expression of different traits in organisms.
Mendel was the person that deduced that genes come in pairs and are inherited as distinct units from one parent. He tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. He also recognized the mathematical patterns of inheritance from one generation to the next, and he created laws of heredity. Mendel did a lot of great work in his experiments, and they took about 8 years to complete. He published his work in 1865, but unfortunately he wasn't really recognized or thought of as important until after his time. We know that he worked with over 10,000 different pea plants during his experiments, and he took meticulous results, recording down all of his data by hand.
All right, and if you have to memorize Mendel's laws, an easy way to remember them is pretend that at the monastery, Gregor Mendel, who was a monk, had a nickname named Sid. I don't know why, it's just part of the story, but Sid will help you remember the three main laws that he came up with: segregation, independent assortment, and dominance.
A gene or genes are pieces of DNA that determine your traits. An allele is just a different form of a gene. If we're writing out genetics problems, this might be written as a big B or a little b, for example. A recessive gene can be masked by a dominant one, and a dominant allele can mask a recessive one. Often dominant alleles are represented by capital letters. A genotype is the actual combination of alleles that somebody has. A phenotype are the physical characteristics that come from that genotype. So for example, your physical characteristic could be tall. That's your phenotype. Homozygous is when we have an allele pair that has two of the same allele. So for example, a little a, or a big A, big A. Heterozygous is when we have two different alleles together in one genotypic pair. For example, big A, little a.
So some basic review for Mendelian inheritance. This is our classical dominant recessive type of inheritance. Remember that genes are often represented by letters, and different versions of genes could be capital or lowercase letters. So here we see the trait for this axolotl. The color of their skin is either going to be pigmented, so this dark color, or without pigment, so this albino axolotl. And to represent these different versions of these traits, we have letters that are going to be different alleles. So the pigment allele will be a big A, and the albino allele will be a little a. Now, each organism has a genotype. These organisms have two genes for a particular trait. So this albino axolotl is homozygous recessive, meaning it has two little a's. So little a would be this axolotl's genotype, but its phenotype, its physical characteristic for the trait, is albino. That's the color. So remember, phenotype, physical characteristic. Genotype is the actual combination of alleles or genes. Now, this pigmented axolotl could be one of two genotypes. It could either be big A, homozygous dominant, or it could be big A, little a, because this big A is dominant over the recessive allele, and it's going to mask or cover up the effects of this recessive gene. So when we see a pigmented axolotl, it could be either one of these genotypes, but both of these genotypes will give us the pigmented phenotype. All right, so let's take a look at a practice problem involving these and our tool, the Punnett square.
So let's say we have two pigmented axolotls that are both heterozygous, meaning they have one big A and one little a, and they are going to mate. We can use things like a Punnett square to see what the chances are of their offspring either being albino or having pigment. So in this case, to set up a Punnett square, we would take one parent and put it on the top of the Punnett square, separating out their alleles or their letters. We would take the other parent and separate out their alleles or letters as well. And then what we do is we drop down all our letters and we fill in the Punnett square. So this big A gets dropped down here and here. This little a gets dropped down here and here. And this big A gets dragged over here. And this little a gets dragged over here. So now we have the results of our Punnett square. We can see that there's a 25% chance of having offspring that will have a homozygous dominant genotype, meaning big A, a 50% chance of having offspring that will have the heterozygous genotype, so big A, little a, and a 25% chance or 1 in 4 chance of having little a, or homozygous recessive. Now, that's a lot of words. What does that mean? So these three combinations here, big A, or big A, little a, that would all mean this axolotl has pigment or is a dark color because A is dominant over little a. So we have 2 copies here. It doesn't really matter. Here we have 1 copy of the dominant trait, so it's going to be dominant over the albino trait. An axolotl with little a, though, that genotype means it doesn't have any pigment because it has 2 copies of the recessive allele. All right, let's get into some practice. Today's practice is not multiple choice. It's going to be some fill in the blank. You might want to get some scratch paper out to work on those Punnett squares. Let's do it. All right, for each of the genotypes below, indicate whether it's heterozygous, meaning 2 different alleles, or homozygous, meaning 2 of the same alleles. Should be pretty easy. You can do it in your head or pause the video and do it on paper. Here we go. All right, so I'm not going to read all of these out loud. You can check your answers here, but I'll do a few of them to go through. Remember, big T, that's homozygous, and technically we call it homozygous dominant to have 2 dominant alleles. Big B, little b is heterozygous. We have 2 different alleles, 1 dominant, 1 recessive. And then let's find this little t here. That is also homozygous, but we call it homozygous recessive since both the alleles are recessive. All right, let's keep moving. Determine the phenotype for each genotype using the information provided about the organisms. So I want you to tell me what the phenotype is, what their physical characteristic is based on the information provided and the genotypes. Ready? Big Y is yellow because it's 2 dominant alleles. Big Y, little y is still yellow because that big Y masks the little y allele, giving us a yellow phenotype. And then little y is blue, the recessive phenotype. You need 2 recessive alleles for that. Gray feathers are dominant to orange, so big G, little g is going to be gray as well as big G, and then little g is going to be orange. All right, for each phenotype, we're going to reverse it, give the genotypes that are possible for this chicken. A red comb, which is the part in their head, big R, is dominant to a gray comb, little r. And then blue body color is dominant to white. So tell me what the possible genotypes are for each of these phenotypes. Ready? So a red comb could be big R, or big R, little r. We could get both of those phenotypes from those 2 genotypes. And then to get the gray comb, we need little r. And then blue body color could come from big B, or big B, little b, and white body color could come from little b. All right, let's put it all together with some Punnett squares. Blue the raptor met Charlie raptor at a dino dance. All hypothetical here. Of course, in the Jurassic Park movies, they're all female, so this doesn't make any sense, but we're just going to pretend. Charlie is heterozygous for his green stripes, but Blue has blue stripes. Create a Punnett square to show the possibilities that would result if Blue and Charlie had offspring. Go ahead and take out some scratch paper and try it on your own, and pause the video, and then hit play when you're ready for the answer. Here we go. So we know that Charlie is heterozygous for his green stripes, so that makes us assume that green is the dominant allele, so that's going to be a big G. And then to get blue, we would have little g here. So if we put it together in a Punnett square, we put one of the parents on one side, another parent on the top, and then we drop down each of our letters to get the following results in our Punnett square. Now it looks like out of the 4 possible combinations, 2 of them can be big G, and 2 of them can be little g. So what are the chances of a child with green stripes? Well, let's look at these phenotypes. Now big G would give us green, big G would give us green, little g would give us blue, and little g would also give us blue. So 2 out of 4, or if we simplify it, 1 out of 2, or 50%. And then chances of a child with blue stripes, it's the same, 2 out of 4, or 50%. time for a fun one. Everyone in Anna's family has dark hair, which is the dominant trait for hair. Her sister, however, has white hair. What are the possible genotypes of their parents? Create a Punnett square to show the possibilities. All right, so take a moment and think about what the possibilities could be for her parents and sketch out a Punnett square of your own. We'll reveal the answer in a minute. Go ahead and pause the video if you need more time, but here it is. So if the parents are both heterozygous for that trait, meaning they each have a dominant allele and a recessive allele, this could produce possible combinations where there's a 75% chance of offspring getting the brown hair trait, but there's also a 25% chance that the offspring could get the white hair trait. Now keep in mind that hair is actually way more complex. There are more than just one dominant and recessive alleles for hair color, and we'll get into that in just a minute. Okay, that was just a very basic practice of some Mendelian genetics, simple inheritance. So I encourage you to go practice more, find some more practice problems, do your homework for class to really get the hang of it. So in this problem here, use the traits from the previous problem. So we're using this yellow allele and green allele for the recessive and the dominant. Two of the offspring have self-fertilized. So we're going to take two of these, and it doesn't matter which two because they're all the same. So self-fertilized just means they're going to cross. So we'll put the cross here as big Y, little y, crossed with big Y, little y. And we can write the key and write same as above in this case because it's the same as this particular trait up here. Okay, so two of the offspring have self-fertilized. Show this in a Punnett square, identify the genotypic and phenotypic ratios. So the way we set this up, we're gonna put parent one here, so big Y, little y, and this is when we wanna be really careful with our big and little letters so we don't confuse them later on. So make sure your capital letters are really capital and your lowercase letters are really lowercase. So I like to do my little y's a little bit differently just so I don't confuse them. So now we're gonna do the cross. We'll bring this letter over here and this letter down here. We're going to bring this little y here and then this big Y down here. And remember, like I said, normally we flip-flop it if the recessive one comes first. We're going to bring this Y over here and then this little y down here. And then finally in our last box, this is how we fill it out. All right, so this is our Punnett square. Now let's look at our genotypic ratio. It looks like we have, as far as big Y's goes, we have one of those. As far as the heterozygous, the big Y, little y, we're going to have two of those. And then for the little y, we're going to have one of those. So we call this a 1 to 2 to 1 genotypic ratio. Now for the phenotypic ratio, let's look at the traits. So if we have one dominant allele, even though we have one recessive one, we're actually going to see the dominant trait here. So this big Y, no question, we know that that's going to be yellow. So as far as yellows, we know we have one here. These are going to be yellow as well because we have at least one dominant trait. So we have one here and then another here, so that's 3 yellows. And then as far as the green, which is recessive, put my messy handwriting here, we just have one green. So we tally this up. This is actually a 3 to 1 phenotypic ratio. So we will write 3 to 1 here, or we could say 75% chance of getting yellow offspring and 25% chance of getting green offspring. Can't write. Okay, moving on. All right, so I've gone ahead and completed the ones in the back, but let's go through and look at the answers. For number 3, it says one pea plant is homozygous recessive for white flowers. You decide to cross it with a plant that is heterozygous red. So now we're going to make our key first. Because red is dominant, we're going to give it the big letter. So big letter R will be red. And then the little r is the allele for white. Now, if from this word problem we can figure out homozygous recessive, that means little r, and then heterozygous red would mean two different. Hetero means different. So big R, little r. So our cross would be big R crossed with little r, and then we're going to set up the Punnett square. Typically, we can put our first parent on this side, our second parent on this side. If you flip them, it's fine. It's going to give you the same results. So if your answers look a little bit different from mine and you have your heterozygous on top and your homozygous recessive on the side, that's fine. We fill it out Punnett square. These are the answers we should get. Our genotypic ratio, we see we have 2 heterozygous and 2 that are homozygous recessive. So we can say 2 to 2, but we always want to simplify. So 2 to 2 actually simplifies to 1 to 1. So that's what we'll write our genotypic ratio as. Now, if you wrote 2 to 2, it's not wrong technically, but this is better. Now, our phenotypic ratio, again, we have 50% chance of getting red and 50% chance that the offspring will have white flowers. Now, the question here is, what is the probability the offspring will have white flowers? So we need to answer that in the space below, and that is 50%. Now, sometimes in genetics problems, it'll just ask for the genotypic and phenotypic ratios. Remember, genes are the actual genotype combinations, and phenotypic ratios, that's what actually physically the traits are going to look like. Now, sometimes it's going to ask an actual question, specifically, what is the probability that the offspring will have white flowers? So what chance do we have that we're going to get offspring with white flowers from this cross? In garden peas, tall is dominant over dwarf. So let's write our key. Big T for tall, little t for dwarf. One homozygous dominant tall plant is crossed with a dwarf. What will be the phenotype of the offspring? Okay, so homozygous dominant, big T, crossed with a dwarf, little t. So we'll put one parent on one side, one parent on the other side, and we're going to get 100% heterozygous, so all big T, little t's. So our genotypic ratio, we can just write 100% big T, little t, or just 1. And then our phenotypic ratio will be 100% tall. So that is the trait for tall because the dominant trait is going to appear in this plant for the heterozygous individual. You have a purple flowered pea plant. You do not know its genotype. Diagram the test cross you would conduct to determine the plant's genotype and what results you would expect if the plant was heterozygous and if the plant was homozygous. And then it asks for the ratios. So a test cross is when you don't know what one of the alleles could be. We know it's purple, so we know it has to have at least one big P, but we don't know what the other allele is. It could be heterozygous with a little p, or it could be homozygous dominant with 2 big P's. And in order to figure that out, we need to use a homozygous recessive individual, and we call this a test cross. So we use someone that we know is little p, and we cross it with our plant. And then the ratios we get will show us what this mystery allele is. So for this one here, we have 2 little p's on this side because that's what the parent is that we know. And then our unknown parent we'd put at the top. And then we would do the cross, we would actually measure and count the offspring, and it would tell us what the actual parent was. So if it actually was heterozygous, this is what the results would be. If it was homozygous dominant, this is what the results would be. So you can figure that out, look at your possible ratios. And then if you were actually crossing these plants and measuring the offspring and seeing what they look like, you could figure out this missing allele. And that's how Mendel actually did a lot of his genetic problems, was through this type of logic. All right, number 3, both parents can roll their tongue, yet their child cannot. What must the parent's genotype be? Rolling of the tongue is dominant. So big R is for rolling, little r is for non-rolling. Both parents can roll, so we know it's big R, little r. Yeah, so we know that they at least have one big R, excuse me, I gave it away. And we know that they have to have each one little r because their child cannot roll their tongue. And in order to not roll your tongue, you need to be homozygous recessive. So in order for the parents to both be able to roll their tongue but also pass this recessive allele onto their offspring, we know they have to have at least one little r each. One of them couldn't be homozygous, for example, so that's why I crossed this out. So we know the parents must be each heterozygous. Okay.
Today we're going to be talking about non-Mendelian genetics, or things that aren't basic dominant and recessive patterns of inheritance. Through this video, we're going to do some content review and then some practice questions at the end. So let's get started. Humans, most human traits are very complicated genetics-wise. In fact, most human traits are polygenic or polygenic, meaning they are controlled by many different genes interacting together. There's very few classic Mendelian inheritance instances that show up in humans. So some of these are actually genetic disorders, like cystic fibrosis
fibrosis, sickle cell anemia, PKU, and we'll talk more about genetic disorders when we get to chromosomes too, which is tomorrow's video. But I just wanted to mention that a lot of examples that we might see or use in biology classes are actually incomplete versions of what's really happening with human genes, because many human traits are caused by combinations of up to thousands of different genes interacting together. And the interactions of different genes can cause differences in phenotypes, and we have a spectrum or a range of different phenotypes in many instances. For example, hair color, skin color, it's not just a yellow pea or a green pea. It's much more complex than that. And then of course we have epigenetics too, where we have different factors that are controlling which genes are turned on and off in which environment. So it does get very complex. But today what we're going to talk about are patterns of inheritance where we don't just have one single dominant allele and one single recessive allele. We can have things such as incomplete dominance. This is when we have a blending of traits. So there may be two different alleles here. For example, the classic one is something like a red flower and a white flower. We'll pretend this is white on the screen. And then if they are crossed or bred, they would generate pink flowers. So let's see how this works. So let's say that in this incomplete dominant scenario, we're going to represent red flowers with big R, white flowers with big W, big W. Notice how these are two capital letters for both alleles. And we're using different letters too instead of R's. That would get really confusing if they were both big R's here. So we set the Punnett square cross up like we would do any classic Punnett square, and we see that our combinations in this case could only be big R, big W, RW, 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 RW. And so what that results in is a phenotype that's a blend of the two parent traits. So RW would actually give us pink, that intermediate characteristic, instead of one or the other, which is really interesting. Codominance is when we have a representation of both traits in the organism. So we're not meeting in the middle, but instead we have both traits show up at some level. Roan cattle are a very common example of this. But with flowers, again, to do it very simply, let's say we have purple flowers and pink flowers. Instead of getting a blend of that purple and pink, we would get some petals that are purple and some petals that are pink, or some spots in the petals that are completely purple and some spots in the petals that are completely pink. So let's see how this works with the Punnett square. In this case too, we use capital letters, two different letters to represent the two different traits. Big P for purple, big B for pink. We cross these two. What we end up getting is, just like we saw in the previous example, these heterozygous offspring that are a combination of both the purple allele and the pink allele. And so what that gives us are flowers that are not a blend of the two colors, but instead show both traits. Blood types we'll see as an example for codominance very frequently. Now, blood types are very interesting. They're controlled by several different alleles. So we have something called multiple alleles that are a possibility here. It's not just A or B. We can have the allele for blood type A, the allele for blood type B, or the allele for blood type O. So that's three different alleles. And then remember, a genotype is two alleles together. So we have lots of different possible combinations here. And AB blood is actually a codominant genotype. So if someone has the allele for type A and the allele for type B, they will represent antigens for both blood type A and blood type B. And then we can talk about really interesting patterns that occur because type O is actually recessive, but A and B are codominant. So that's a good example of codominance. And then of course there's the Rh factor and lots of different things we can talk about with the different blood types and who's a donor and whatnot. Here would be how you would set up a cross for a type A homozygous individual and a type B homozygous individual. So we would put their genotypes on either side of the Punnett square, and it looks like if these two individuals crossed, we would have 100% chance of their offspring ending up to be type AB blood. Let's see it again with an individual that has a genotype for type that has type B blood, but they also carry a little I allele. Same thing over here. They have type A blood, but they carry that little I allele. And in this case, with this cross, we have a 25% of these two parents having kids with type O blood. And we noticed that they had the same phenotypes, so the same type A and type B in this situation, but because the genotypes are different, we get different results in the potential genotypic combinations for the offspring. Let's move ahead now to talk about sex-linked traits, which is another different type of inheritance that we see in humans. So these are traits that have alleles that are located on sex chromosomes. So in a human karyotype, there are 23 pairs, so 46 total chromosomes, and 2 of those are sex chromosomes. In biologic females, it's an X and an X. In biological males, it's an X and a Y. If something is an X-linked trait, that means it appears on the X chromosome. Generally, there's going to be more X-linked traits that we'll talk about just because the X chromosome is much bigger and there's more space for traits. Colorblindness is a very common X-linked recessive trait, as is hemophilia. That is also X-linked recessive. And Y-linked traits, there's fewer of them that we'll study and talk about just because the Y chromosome is smaller, and many Y-linked traits actually lead to sterility. So if someone had one of those Y-linked traits, they could not pass it on to their offspring, because if someone is sterile, they cannot have offspring. All right, so let's just take a look really quick. Remember, females have the XX chromosome pair and males have an X and a Y. If we look at a human karyotype, so 1 through 22, these are all the autosomes or the non-sex chromosomes that humans have, and then the 2 sex chromosomes are either XX or XY, and so this is a karyotype of a human male. So let's take a look at the inheritance for color blindness. So the allele for normal vision is dominant, and the allele for color blindness, or someone who is affected with this color blindness trait, is recessive. So we'll show that with a little h. Here we show 2 normal individuals. Neither of them carry any of the genes for color blindness. In this case, this female is a carrier here. She is still normal though, because she has one dominant allele, that big H, and that can mask or cover up the little h. So she is still fine. She has normal vision. She's not colorblind. The male here is normal as well. Now in this case, the female is colorblind. She has 2 of those alleles for colorblindness. They're both carried on the X chromosome. The male is also normal. And here we have 2 colorblind individuals. We have a colorblind female and a colorblind male. But notice with the male, he only needs 1 copy of that recessive allele in order to be colorblind, because he only has 1 X chromosome. So there's no dominant allele present to be able to mask that other allele, and so he will be colorblind with that 1 copy of the affected allele. And here we have a case with a female who is normal. Her alleles are unaffected and the male is colorblind. All right, so how do we set up a Punnett square for a sex-linked cross? It's not overly complicated. We just have to make sure we carry the X's and the H's or whatever letter we're using for the trait together. We don't separate those, but we are going to separate each individual sex chromosome. So if we were to cross these 2 parents, we would set up the Punnett square like this, and we make sure we write the letters as like an exponent, like it shows in their genotypes up at the top. And then just like a regular Punnett square, we'll carry down the letters, bring them across to see the possible combinations for the offspring. So in this case, we could have, out of all the possible offspring, there's a 50% chance that this couple will produce females who are carriers for color blindness but not have color blindness themselves, and a 50% chance that they will have offspring who are males who are normal. They don't carry any of the colorblind alleles. So remember, females can be carriers of sex-linked traits because they have 2 X chromosomes, and technically in biology, a carrier is someone who does not display the trait, but they could pass it on to their offspring. So in this case, a carrier for color blindness would look like this, and males technically cannot be carriers of sex-linked traits. So obviously someone who has the trait could pass it on to their offspring, but a carrier, if we're going by that strict definition, males can have conditions that are sex-linked, but they're not carriers technically. All right, let's get into some practice. Get out some scratch paper. We're going to be doing some Punnett squares, and if you want to pause and take the time to do these on your own or mute me and go at your own pace, you are more than welcome to do that. All right, our first cross, we have a colorblind mother and a normal or unaffected father. These are their genotypes. Go ahead and give the potential genotypes and phenotypes of their offspring. And when you're ready, here they are. We have a 50% chance that they will produce carrier females, so X big H, X little h, and a 50% chance of having colorblind males. So in this particular couple, if they have a boy, he will definitely be colorblind. That's the only possible outcome. All right, second cross, carrier mother, colorblind father. These are their genotypes. Go ahead and cross those parents. And when you're ready, here is our outcomes. So we have a 25% chance their offspring will be a carrier female, 25% chance their offspring will be a normal male, 25% chance their offspring will be a colorblind female, and a 25% chance their offspring will be a colorblind male. So lots of different possibilities in this particular cross when we have each parent carrying 1 allele for the colorblind trait. first, a little bit of vocabulary. Some students mix up chromosomes, chromatin, centromeres, sister chromatids, so we're going to go over that right now. First of all, this is a very bad illustration of a chromosome, and it's kind of representing a misconception that a lot of students have about what chromosomes actually are. The DNA in a chromosome isn't actually just one strand with, a couple dozen bases. In fact, it is millions of bases. Many chromosomes contain 50 million nucleotide base pairs, some even 300 million nucleotide base pairs. So what we're seeing here is a very bad representation of a chromosome. Remember, chromosomes are DNA that are tightly condensed and packed, wound up around specific proteins called histones. So let's talk a little bit about that organization and then we'll get into the vocab. So if we have our DNA molecule, we know that in the middle it's connected with base pairs, there's a backbone. When it starts to be wound up, it is wound around these protein complexes that can unwind to be transcribed or expressed. And when it prepares for cell division, it has to be very tightly wound into these structures called chromosomes. So that winding of the DNA around these proteins is going to be very important for us to organize the DNA in a way that allows it to be distributed evenly into the daughter cells. So when it is in the nucleus and it's not going through mitosis, it's not wound compactly into chromosome forms, we call it chromatin. Chromatin is just that complex of DNA and protein. Sometimes it's shown here, you can see it kind of tiny, it's sort of this like spaghetti-like substance. Now, when it prepares for cell division, the DNA, and you can kind of see it here, will condense into those chromosomes. In humans, we have 46, so it'll condense into those 46 individual chromosomes. Remember, these are long, long strands of DNA that are being packed tightly together. And before it goes through mitosis as well, the DNA has to duplicate. So each chromosome actually has two identical sister chromatids. They're the same on each side. And in mitosis, those chromatids are going to split apart. So let's talk about that vocab one more time. This whole thing is a chromosome, and you can see the individual chromosomes in the nucleus, but when it's not condensed into the chromosomes, we call it chromatin, or just that bundle of DNA and proteins is chromatin. The chromosome is attached at the center by a centromere, so centromere, center. And then each side of the chromosome has an identical sister chromatid. So one section here, that's one chromatid, and one section here, that's one chromatid. So one more time, chromosome, chromatid, centromere, and chromatin here in the center. Okay, we're going to be referencing these throughout today's reviews. I just want to make sure you had them all straight.
Genes can be influenced by the environment. For example, in identical twin studies, we can see that identical twins separated at birth can have different heights and weights after 20 years. Most likely the cause of these differences is that each twin was provided a different diet and had different physical activities throughout their lifetimes, because an environment does play a role in influencing individuals. So let's look at a karyotype. Now remember, a karyotype is just an image of all the chromosomes in the nucleus of an organism's any one cell. So in humans, we have 46 total chromosomes, or 23 pairs. So remember that the first 22 pairs, these are called autosomes, and the two that are left, these are our sex chromosomes. If you're biologically female, you have two X sex chromosomes. If you're biologically male, your sex chromosomes are X and Y. Now we do see some differences in certain genetic disorders where we can have different combinations of these, but we'll get into that when we start talking about genetic differences. So how do we get a karyotype of a baby that's not born yet? Well, we can use something called amniocentesis. Amniocentesis, or an amniotic fluid test, is a medical procedure used in prenatal diagnosis of genetic disorders and fetal abnormalities. So what happens is a small amount of amniotic fluid, which is here, is going to contain some fetal tissues. And it's going to be extracted from the amniotic sac with a large needle. And once we take it out, the DNA can be examined for genetic abnormalities, or we can simply see the biological sex of the individual. Now, not all women choose to do this because there is a small risk involved in the procedure of amniocentesis, but if you elect to do it, or if you're high-risk pregnancy, some women will be able to see a karyotype of their child before they're born. Now remember, a normal human karyotype has 23 pairs of chromosomes, including the sex chromosomes here. This individual has all 23 pairs, one X and one Y. If you look closely, you can see this individual is a biological male. So when we look at our karyotype, we're going to identify and evaluate the size and the shape and the number of chromosomes in our sample body of cells. Some problems that we could see would be duplicates of chromosomes where there shouldn't be, or deletions of certain arms of chromosomes, or translocations of chromosomes.
Now, different events in meiosis where the chromosomes don't split apart the way they should can lead to different chromosomal conditions, meaning that in those egg cells,
and after fertilization, we have a variation on the number of chromosomes that we would typically see in human cells. So you might have heard of something called trisomy 21 or Down syndrome. That's when we have 3 copies of the 21st chromosome instead of 2. And these extra or missing chromosomes can have varying effects on humans, from a variety of symptoms to having huge effects on the organism. These are what are called karyotypes, which is where we line up all of the chromosomes for a picture in different organisms. And so a typical biological female will have 2 X chromosomes, and a biological male will have an X and a Y chromosome. So there are different chromosomal conditions which can vary, and sometimes we see XXY or just a single X. XYY, there's a lot of different variations. So here's a look at trisomy 21. If you look at that 21st chromosome, we see 3 copies.
Now, chromosomes aren't everything and genes aren't everything. The environment has a huge influence on phenotype or on the resulting traits of an individual. Some scientists estimate up to 75% of who you are and how your traits operate is due to environmental factors. We're learning so much more about epigenetics and how different things can turn genes on and off in different ways. So, environmental factors can influence gene expression and lead to phenotypic plasticity. This is when individuals with the same genotype have differences in the phenotype that they express because of environmental conditions. For example, there's some mammals like rabbits that can exhibit different fur colors and different temperatures. Hydrangeas are another popular example of this. The petals of certain hydrangeas can change color depending on the pH of the soil that they're planted in. So, for example, here we see that blue flowers are going to appear when the soil is highly acidic, and pink flowers are going to appear when there's a neutral or slightly acidic soil. Many reptiles, such as crocodiles and turtles, have sex determination based on temperature because sex determination in these animals is highly dependent on hormones, and these hormones can be changed by the activity levels of different enzymes depending on a specific temperature. So we can see an entire brood of turtles hatching to be one sex because they were all incubated at a particular temperature. We know that some genetic differences in individuals are due to single allele or single gene changes, and others are due to changes in entire chromosomes. A lot of times this happens because of non-disjunction or when a chromosome doesn't separate properly during meiosis. A lot of genetic disorders we call autosomal because they're occurring on any chromosome that's not a sex chromosome. So all the chromosomes except for sex chromosomes, remember, are autosomes. And so autosomal conditions are caused by mutations on chromosomes that are not sex chromosomes. You might have heard of Huntington's disease as an example of an autosomal condition or an autosomal dominant genetic disorder, which often doesn't appear until later in life, so people can pass it on to their offspring without even knowing that they have it. And then there's lots of autosomal recessive disorders like Tay-Sachs disease, cystic fibrosis, PKU, sickle cell anemia. Chromosomal conditions are usually caused by non-disjunction, which means when those chromosomes are supposed to separate, especially during anaphase 2 of meiosis, we're getting an extra chromosome ending up in the sex cell. So things like trisomy 21, Turner syndrome, Klinefelter's, poly-X, Jacob syndrome, these are all examples of chromosomal conditions. So let's jump into a few practice questions. Starting here, 2 identical flowering plants of the same species are planted at separate locations in a garden. Plant A produces green flowers and plant B produces pink flowers. When the soil is tested, the pH near plant A is found to be more acidic than the pH of the soil near plant B. What is a likely explanation for the difference? Think about it, and you can pause and go ahead and mute me if you want and go through these questions at your own pace. Remember, do what is best for you in your learning process. Correct answer is C. The soil pH influences phenotypic expression. It's not changing the genotype, it's changing the expression of the phenotype. 2. Genes are inherited, but their expressions can be modified by the environment. This statement best explains why A. Some animals have dark fur only when the temperature is within a certain range. B. Identical twins who grow up in different homes have the same characteristics. C. Offspring produced by means of sexual reproduction look exactly like their parents. Or D. Animals can be cloned, but plants cannot. Correct answer is A. Some animals have dark fur only when the temperature is within a certain range. Again, that's the environment influencing the expression of those genes. Seedless watermelon have triploid cells. This makes it difficult for the plant to successfully produce seeds. Why? A. Triploid cells hinder even chromosome pairing in meiosis. B. Triploid cells cannot proceed through the S phase of mitosis. C. Triploid cells increase chromosome stability in meiosis. Or D. Triploid cells have fewer chromosomes than diploid cells. Think about it. Correct answer is A. Triploid cells hinder even chromosome pairing in meiosis. This graph shows the effects of temperature on sex ratios of the T. scripta, the pond slider turtle. If eggs are incubated below 28 degrees, what will be the result in the brood of turtles? So we have a graph here. I'm going to take my video off so you guys can see this a little bit better. And let's go through the answers. A, all the turtles will be male. B, all the turtles will be female. C, 30% of the turtles will be male. Or D, none of the turtles will survive. Think about it. Correct answer is A, all the turtles will be male. So we can see from the graph that at 28 degrees or below, we have 100% male turtles coming up from this environmental condition. But temperature will have an effect on the expression of some of those genes and the resulting hormones to determine the sex of the different turtles.
All right, so now we're going to talk about how we can trace the inheritance of traits through several generations. So a pedigree is a special type of chart that depicts family history or the inheritance of a specific trait. And we have different symbols in our pedigrees to show us different information. So squares are used to indicate biological males in a family. Circles are used to indicate biological females. If a person is affected by the trait, whether it's dominant or recessive, we're going to track that by shading or coloring in the shape. So a line between a male and a female indicates a marriage or a union, but a line drawn down from that marriage or union indicates offspring, so that they're together and they've had offspring. Sometimes you'll see shapes shaded in halfway, and this indicates usually a heterozygous carrier of the trait. And we won't see that every time in a pedigree, but we'll get to some examples in a little bit. As my students know, I was a big Star Wars fan. This is me dressed up as Rey a few years ago. But let's practice pedigrees by tracing the inheritance of a hypothetical trait that could have been inherited in Star Wars. So let's assume the power of the Force is inherited as a recessive trait. So somebody with the Force would be little f, somebody without the Force could be big F, and someone who carries the Force could be big F, little f. So they don't present the Force really, they don't have the powers, but they could pass it on to their offspring. So Star Wars people, don't get too mad at me, but Anakin Skywalker has the Force. He marries Padme, and they have twins, a boy, Luke, and a girl, Leia. Both children have the Force. Luke does not have children as far as we know, but Leia means a normal pilot, meaning he does not have the Force, Han Solo, and they have a child, Ben, who does have the Force. Draw a pedigree showing the inheritance of the Force in this family. So let's see what we can do here. So remember, somebody with the Force is little f, someone without the Force definitely is big F, but somebody who could carry the Force is big F, little f. So go ahead and pause the video and see if you can come up with a pedigree for this specific family. All right, let's see what we got. So this is what I came up with here. There are some variations you could do. You might not want to do the half shade-ins for carriers. Another symbol on pedigrees, if you get to more complex ones, you might see twins depicted like this, but we're going to leave it like this for today. Okay, so up at the top we have Anakin and Padme. We know that Padme has to be a Force carrier because both of her children end up having the Force, little f, little f. Now, here we're talking about Leia. So Leia obviously has offspring with the Force as well. So because this offspring is little f, we can assume that Han Solo was also a Force carrier if it works this way. So there we go with all our characteristics. Now we know that the Star Wars lore doesn't always work that way, and if you wanted to try to trace out some other family members, be my guest. But remember that this is just one way to track inheritance through a family. Here's some real quick hints for analyzing pedigrees. So if the individual is recessive, then both parents must have at least one recessive allele. So parents are either heterozygous or homozygous recessive. If an individual shows or presents with the phenotype, then at least one of the parents must have the dominant phenotype. And this will be pretty obvious when you look at the pedigree. If both parents are homozygous recessive, then all of the offspring will also be homozygous recessive. Now, you might be studying genetic conditions or genetic disorders that are caused by different types of alleles, like recessive or dominant alleles. So an extension option right now for this lesson would be to go and do a little bit of research to find 2 genetic disorders that could be caused by recessive alleles and 2 genetic disorders caused by dominant alleles. If you want to pause the video and go take care of that, go ahead. Thanks so much for watching our biology makeup lesson in genetics. If there are other topics that you're interested in, please let me know in the comments below. I hope this video has been helpful with your studies or your review. Give it a like if it has, and be sure to share it with a friend. I'll see you later.